All right, today we're going to talk about mutations. Once again, these notes are given in class, so you should only be watching this video if you were absent in class and didn't get the notes, or if you wanted to brush up on mutations after you listen to the notes in class. So we've covered the cell cycle, we've covered how cells divide, we've covered how cells replicate their DNA, we've covered how cells make proteins. But now we're going to cover what happens when there are changes in the DNA strand. So a mutation is what happens when changes happen to the DNA strand. And what a mutation is a permanent random change in the order of the DNA bases. That could either be due to mistakes happening when the DNA is copied over, or it could be the result of environmental factors such as carcinogens, which we talked about a little bit before. So like UV radiation would be an example of a carcinogen, or chemicals or something like that. When a mutation happens in the DNA strand, protein synthesis is going to make a protein that is not exactly the same as whatever the DNA strand originally was, because protein synthesis goes off the DNA strand as it currently is. So if there's an error in it, there's going to be an error in the resulting protein. Mutations are, though, part of what contributes to genetic variation within a species. It's not always bad. So if you've ever seen X-Men, they talk about mutations, mutants, and stuff all the time, and that's exactly what I'm talking about here, mutations in the genetic code. But unfortunately, real-life mutations do not cause superpowers. Most of the time, mutations, when they happen, don't really do a whole lot because there's a lot of DNA that we have that really doesn't code for much, or those genes are switched off. But when mutations happen in important genes, those can result in different traits, or sometimes they can result in bad things happening. But a mutation could be inherited. So like, let's say the DNA code of somebody's ancestor changed randomly, and then that ancestor was able to pass on that change to their offspring and so on and so forth. And this happens when the mutation creates a positive effect quite often, because positive mutations tend to help a species survive better. And so if that happens, then that mutation will be passed on because the offspring will survive. And then you'll see that mutation more and more in a population. But also, like I said, they could theoretically disrupt normal gene activity and cause diseases. So mutations, when they happen and result in a negative effect, they can cause cancer or they can cause death, depending on what it is and where it is and how serious it is. So there are two different main types of mutations. So there's gene mutations, which are changes to a singular gene, like one gene that codes for one thing. And those gene mutations are split up into point mutations, which are changes at a single point of the DNA strand. So up on our top strand here, we see that our sequence is CTGA-GT, but when our point mutation happened, that A was changed to a C. So now our strand reads CTG-CGT. Okay? This doesn't always have to be one nucleotide. It could be a few nucleotides. So an example of that would be take our original strand and then maybe change this one to a C instead. So multiple nucleotide point mutations can happen in the same DNA sequence. It's not always just one. You could have a substitution point mutation, meaning one letter is substituted for. So that's the situation that I just showed you when that A turned itself into a C. So when that happens, we have a change of, usually it's just one amino acid in the resulting protein. So if we look at our original strand up here and our mutated strand down here, we have our original sequence of DNA, TAC, CGA, TGG, AAA. We have our mRNA strand that results from that, and the amino acid sequence methionine, arginine, threonine, and phenylalanine. But when that C here in the original strand changed to a T instead down here, now our mRNA strand is different by one letter, but that resulted in a change of amino acid. So instead of methionine arginine threonine, it now reads methionine histidine threonine. So that could be a problem, or maybe it could not be a problem. Sometimes when amino acids are substituted out, it doesn't change the protein a whole lot, but sometimes it does. It honestly depends, case for case. And if that doesn't make a whole lot of sense to you, you can also think about point mutations like this up here. Our original sentence reads, the fat cat ate the wee rat, and the substitution of C turning into an H now makes the sentence, the fat hat ate the wee rat. So that doesn't make a whole lot of sense. It changes the meaning of the entire sentence, but it was just one letter substitution. So that's what I mean when I say that sometimes substitutions can change a lot, but if that were a different sentence, one letter difference may not have made an entire difference to the entire sentence's meaning. Okay, so next, you could also have a frame shift mutation. These tend to be more serious than point mutations because they affect a lot more of the gene. And what I mean by that is a frame shift mutation, it shifts the reading frame. So if you look at this sentence, this original sentence again, the fat cat ate the wee rat, if we delete the letter H out of that, but we're still reading in sequences of 3 letters, suddenly you have something that doesn't make any sense whatsoever. We have tef, atk, at, tet, her, and then these 2 floating AT letters at the end. So technically most of the letters are the same, yeah, but this is unintelligible. Like, we have no idea what this is supposed to mean just looking at it. And the original sentence is only 1 letter different. So that's the danger of frame shift mutations. Those could be for one of 2 reasons. Either one letter could have been inserted where it's not supposed to go. So in this top situation up here, in between the A and the G, a T was placed. So now it's going to change the reading frame by adding 1 extra letter, so the codons are going to be different. Or it could happen because 1 letter was deleted, so in this bottom situation, the letter A was deleted, and again, that shifts the reading frame entirely, making that gene unintelligible. So here's another way to look at it. With our original sentence, if you like looking at it the sentence way, still the same, the fat cat ate the wee rat. We inserted the letter A, and now the rest of the sentence after that insertion no longer makes any sense whatsoever. The DNA way to look at it, what actually is going on in your body, we have our original DNA sequence and our mRNA and our series of bases, but when you have now a letter inserted here, this A was inserted when it doesn't belong, now everything after that is suddenly different. And so these ones, more than point mutations, end up causing non-functional proteins a lot more. Here's a deletion example. I'm not going to explain it again because I think you understand how deletion works at this point. You know what delete means. We're deleting a letter. But again, you still get the same effect down here in the gene. Now that we've deleted a letter, the entire thing is now different once again. So frame shift mutations change every single amino acid, and insertions / deletions are frame shift mutations. So if I wanted you to take one thing away from this lecture. It would be the types of mutations and what qualifies as that type of mutation. So point mutation, substitution, frameshift mutation, insertion or deletion. Okay. Chromosomal mutations, on the other hand, are a little bit different because a chromosome, if you recall, is made of many genes. So before we were talking about singular gene mutations. Here we're talking about changes of a whole entire chromosome, which could be like hundreds of genes or thousands, depending on how much is changed. Okay. So we can still experience a deletion mutation in an entire chromosome, except what is deleted is not one nucleotide, it's many genes out of the chromosome, making that chromosome smaller by a lot. We could have a duplication where one section of the chromosome is copied. So here in the original chromosome, you'd see that that little blue section was copied over twice. So now your chromosome is longer. So it's kind of the opposite of a deletion. You could have an inversion where genes switch places in a chromosome or change direction or something like that. Or you could have something called a translocation. And that happens when a part of one chromosome breaks off and it attaches to another part of a different chromosome. So if we look at our original chromosomes in this little situation over on the left, we have purple, red, green, orange, blue, and then green, orange, blue, purple. Over here in our mutated chromosomes, now we have purple, green, orange, blue, green, orange, blue, red, purple. So that is completely different, and that is because genes are breaking off and switching places. And so that's not great when that happens. That causes a lot of issues. So what I want you to take away from this part, again, is the types of mutations that can happen in different chromosomes. So singular gene mutations, we have point mutation substitutions, insertion or deletion. Whole chromosomes, we have deletion, duplication, translocation, and all that. So hopefully you'll hear this lecture in class. If not, hopefully this video was helpful. If you are still having trouble, please talk to me in class.